Hello, everybody. My name is Michael Yabsley, and I'm a professor at the University of Georgia. And I'm honored to have been asked to speak to you guys today about some of the research that we've been doing here at UGA to understand the natural history of guinea worm and to assist with the guinea worm eradication program. So just to set the stage, primarily what we'll be talking about is some work that we've been doing in Chad, Africa, which is in north central Africa. And here's a beautiful picture that shows one of our study sites along the Shari River in Chad. And our story will revolve mostly around domestic dogs and their new role as host for the human guinea worm. And this is the human guinea worm that we'll be talking about. And so this particular parasite forms these blisters that you can see here. And then these long worms emerge from their host, as you can see here. But I do want to back up and just give you some introductory information about this group of worms. So they are parasitic in the genus Dracunculus. Females are famous because they can be up to 3 feet long, quite large. But on the other hand, the males are very, very small and very hard to find. So here you can see an individual animal, both a male worm on the left and a female worm on the right being wound out. So this particular genus has quite a number of species that have been described formally within it. On the left, you can see that there are 4 different species described for mammals, including Dracunculus medinensis, the human guinea worm, and then Dracunculus insignis is a parasite I'll actually talk about quite a bit during this talk. It's a North American parasite that infects a diversity of wild mammals, as well as domestic dogs and cats. And we study this parasite mainly because it serves as a useful model for understanding what's going on with guinea worm. Then on the right, you can see there's actually quite a number of species that have been described from reptiles, mostly from snakes. And so while my talk actually revolves around work to try to eradicate a particular parasite, I understand the whole point of this conference is that biodiversity matters, and it certainly does. And so this is where I can sort of bring in some information about how studies on Dracunculus and their natural hosts all around the world have really been useful in informing some of the studies that we are now doing with human guinea worm. So without understanding the diversity of these parasites, without understanding their life cycles, we really would be taking many steps back to sort of think about the types of projects that we would want to do to understand human guinea worm. And along those lines, although there's a number of species that have been described, the diversity is actually much higher than that shows. There's lots of different worms that have been removed from snakes and likely represent new species. Dogs in Argentina have a species that's actually likely unique. Some work we did a couple of years ago here in Georgia, where Dracunculus insignis is very common, we found that one particular worm, you can see here in the middle, phylogenetically was quite distinct from all the others. And so there was a novel species of worm right here in our backyard, essentially. The next year, a parasite very closely related to that Virginia possum parasite was found in a domestic dog in Spain, which represents the first report of a mammal infected with Dracunculus in Europe. So we don't really understand where that particular parasite came from. And then most recently, there was a human in Vietnam infected with a Dracunculus that presumably is novel. So bringing it back, though, to Dracunculus medinensis, the guinea worm. It causes dracunculitis or guinea worm disease. It's sometimes called a fiery serpent because of the extreme burning pain that accompanies the development of that blister that you'll see in a minute. This parasite's been known for a very long period of time. It's been found in mummies. It's been depicted in old paintings. So pretty long history. The good news is this parasite's rarely fatal, but unfortunately infections are very painful, often debilitating, and this can last for weeks or months. There are possible other complications such as secondary bacterial or fungal infections. These parasites can migrate to unusual locations where emergence may be very complicated, or they may calcify there, die and calcify, resulting in pain or limited mobility in those areas. And then finally, larvae could be released from females that fail to emerge, which could result in an abscess, which could then ultimately lead to other complications. So this parasite has been referred to as a disease of empty granary. And what that refers to is this long-term disability that people have. So they're unable to attend school. They can't go to work or farm. And that all results in economic struggles and food insecurity for these people. So it's certainly a parasite of interest for removing from the human population. There is no specific treatment. The historical way of wrapping this parasite around a stick or sterile gauze is still used today. In addition to that, though, submerging the foot or arm or wherever the blister may be into water to help that worm emerge from the blister prior to it getting into natural water sources is also used. But even there, you can see in this middle picture on the bottom, only a portion of the worm will emerge initially. Then you have to start slowly removing that worm over time. So there is no vaccine and there's no antibiotic treatment available. So I want us to take a step back and just talk about some of the history around the life cycle, since a lot of what we'll talk about today is how we've sort of recognized new transmission routes. The initial transmission route, which is the use of a copepod intermediate host, was recognized all the way back in the late 1870s. There was work done on a related fish parasite that used copepods that essentially suggested to workers that copepods might be an intermediate host. So Fenchenko, who was working in Turkestan at the time, showed that this parasite actually could be transmitted into copepods. So he published his work and unfortunately died soon after that. But the work was picked up by Mason. In 1895, he showed that if you took water and put it on a blister, that it would essentially burst and larvae would come out. And then he too confirmed that when those larvae were fed to copepods, they became infected and matured. There were still naysayers all the way back in the day there. You can see on the right-hand side, doubt whether or not these Dracunculus, when ingested, would actually survive passage through the GI tract and result in infection. So while most people were sort of coming together around this drinking water association, there were the naysayers. Then in the early 1900s, Leeper was working in Ghana, and he essentially showed that you could take larvae and feed them to copepods. They would mature and survive for several weeks. And then he completed the life cycle by feeding those copepods to a vervet monkey, where they developed. Unfortunately, he left Guinea worm to work on other important parasites as well, so he didn't do any more work. But Turkett in 1913 actually then further confirmed the life cycle, this time in humans. Sort of frowned upon now to do that kind of work, but he did show that you could transmit it to people through drinking water with copepods. So the life cycle, in terms of parasites, relatively simple. So you've got that female that emerges from the leg or extremity and releases larvae into the water where they're ingested by copepods. They molt twice to the infective stage. And then if that water is ingested by a human or other appropriate definitive host, those parasites will break out of the copepods, migrate into the abdominal cavity where they mature, mate, and then the female goes on and migrates to subcutaneous tissues where, unfortunately for people like me who study this parasite and both in the lab and the field, it can take 10 to 14 months for completion of the life cycle. So that female remains in the subcutaneous tissues where she further matures and starts to produce larvae. And then at the end of this period of time, generally around 12 months, she emerges and releases larvae for the parasite's life cycle to continue. So in terms of control efforts, in 1986, the World Health Organization and the Carter Center got together and formed the Guinea Worm Eradication Program, and they work with endemic countries to try and eradicate this parasite. So they have developed a number of interventions that have been put into place. A big one is education, just teaching people how this parasite is transmitted so they understand better how to break the life cycle. Another big component of this is the provisioning of safe water sources, because if you are reliant on water sources potentially contaminated by copepods, you're not going to be ingesting guinea worm. In times when you have to get water from natural areas, the use of cloth filters or field life straws, which has a filter inside, essentially just filter out those large copepods that may be infected with guinea worm. And then if you've got water bodies that you know have been exposed or suspected to have been exposed to guinea worm, you can use a bait to kill those copepods. And then finally, there's a large network out there for active surveillance and case detection. And when cases are detected, Folk can be brought into containment centers where you can do that controlled immersion that I talked about earlier so that there's less risk of contaminating natural waterways. So this campaign has been extremely successful. In 1986, when it was begun, there were 22 countries with endemic transmission of guinea worm, and there were about 3.5 million cases a year. Those numbers have steadily dropped over time to just 27 human cases in 2020. And then this year, up until August, there have only been 10 human cases, which is fantastic news. These cases are distributed just across a few countries, Chad having the most cases, both in 2020 and in 2021, and in Ethiopia having 1 case this year, but 11 cases last year. South Sudan has 2 cases this year, and Mali has had 1. Angola also is considered endemic and had a case last year. And then Cameroon had a case last year, but it was highly suspected to have been a cross-border transmission from Chad. So again, because Chad has had the bulk of human cases recently, and as you'll see, domestic dog infections as well, much of our work is going to focus in on Chad. So historically, Chad actually was ruled guinea worm free back in 1998. It was considered no longer endemic in the country. But then in 2010, a human case was detected. And then by 2012, there were dog infections being diagnosed. So dog infections are nothing new. In fact, guinea worm infections in dogs as well as other animals have been reported throughout history during the time that guinea worm was common in these countries. But when guinea worm was eradicated from those areas, dog infections essentially disappeared. And so it was not being sustained within dogs. That situation has changed, at least up until recently. Numbers of dog infections have increased in Chad. And so you can see here, just simple, each year, there was increased recognition of the number of cases in dogs. And because of that, there were a number of new interventions put into place to try to control transmission and infections in dogs. And those included tethering dogs so that they wouldn't have access to waterways to contaminate them if they had a worm. A reward system similar to humans, where if you turn in an infected dog, you would get a reward, and that way that dog could be tethered or contained to prevent further contamination of the environment. The use of a bait in and around villages with known dog infections to try to remove any risk of guinea worms being present in copepods in the water. And then, as you'll see later on, some of the research that's been done sort of suggested fish are involved in transmission. And so there's new interventions in place to get villagers to bury or burn the fish entrails so that dogs and cats and other animals don't have access to them. So Chad last year had almost 1,500 infected dogs, 61 infected domestic cats, and then 2 wild cats. Mali also has infected dogs. Last year they had 8. And then in Ethiopia, there are reports of infected dogs and cats as well. But in Ethiopia, there's actually reports of infected baboons, which haven't been reported in the other countries. So whenever we have an instance like this where a human-specific or presumed human-associated parasite is being found in animals, especially in increasing numbers, the first big question is going to be, is this even the same parasite? The hope would be that you've got 2 different related parasites that are co-circulating in an area, but one is specific to animals and one is specific to people. And so you wouldn't have co-transmission between those 2 groups. So this question has been looked at genetically. And so I know this is a very busy slide and you can't necessarily see these 2 phylogenetic trees easily. But the take-home message here is that the different colors represent different countries for the worms, and the different symbol shapes are different hosts. So whether you looked at full genome sequencing that's shown in the top left, or sequence analysis of a single gene target in the middle on the bottom, you can see that there's general clumping of the colors by country, with the exception of many worms from Chad. Some of them tend to pop up in other locales as well. And then the other big take-home message is that those individual hosts are scattered throughout the tree. So these are all Dracunculus medinensis, and they're using different hosts regardless of the geographic origin. And so this confirms that, in fact, this is the same parasite that's being transmitted between different hosts. Liz Teal also did some really fantastic molecular work using microsatellites to show the same thing. Essentially, different hosts in the same country were infected with the same genetic types of the parasite. So the big question, of course, is why are we seeing infections in dogs increasing? And why now? Why are there so many of them, whereas historically they went away? And why are we seeing increasing numbers in cats and in some places in wildlife species? So if we look at this map of Chad, and the Shari River is shown there with villages with infection and dogs shown in orange, you can see that there's infections going all the way from the north of the Shari all the way to the south in the Sar region and along some of the tributaries as well. And then those black dots show that there are hotspot villages where we tend to see increased numbers of dogs than in some of the other areas. So this is an infection that's widespread across a very large geographic area in Chad in dogs. So knowing that and knowing that human infections right now are very rare in Chad, and there did not seem to be an epidemiological link between dog infections and human infections, we thought there must be something else going on in terms of transmission risks between dogs and humans. So we're thinking, what are we missing? So one of the first things that people started to think about was whether or not there was an aquatic paratenic host involved. So just to remind you of the life cycle, you've got the intermediate cyclopoid copepod host that's infected, and it's ingested by a definitive host during a drinking event. So the thought here was that maybe there's a paratenic or transport host that's ingesting copepods and essentially serving as a bridge host to then be ingested by an appropriate definitive host, leading to infection. And when we start to look around and think about the possible routes via fish or frogs as potential aquatic hosts, we certainly see many opportunities for dogs and cats to have access to fish, either through small fish that are captured during fishing events that may not necessarily be consumed by the people, or when people are cleaning the fish, and all of those entrails and organs that are removed from the fish potentially could be offered to dogs or cats to eat, or at least be left available where they could eat them. And so we can postulate that there's potential chance of transmission via this route. And so when we think about frogs and fish seems logical because of all the fishing activities. But even with frogs and tadpoles, so the amphibians, there's precedence. So some older literature, older studies that looked at transmission of Dracunculus and cygnus and even some of the reptile species showed that they can infect tadpoles. So in terms of human infections, dogs are eating frogs, of course. Humans eat frogs as well, but most of those are cooked, we presume. And so maybe frogs aren't necessarily important for human transmission, but certainly could be involved for dogs. So one of the first things we did was an experimental trial just to see if we could experimentally infect either frogs or fish. And in this particular study, using Dracunculus medinensis, we found that green frogs were susceptible to infection, and we found larvae in their tissues. And then two species of fish that we looked at actually did not become infected. So we headed off to Chad to look for this parasite in aquatic hosts. And we had multiple villages shown here in these purple stars that we sampled aquatic hosts from, which essentially meant catching the animals, grinding them up, using a Behrman-style technique to accumulate larvae from their tissues, and then looking for them under microscopes. So our first trip to the south of Chad and the Sar region was quite successful. We had a single frog during that trip that was confirmed to be infected with Dracunculus medinensis using molecular techniques. So we had 4 more trips after that. So this is all of the fish data, and I don't want you to focus in on all of the details of this table, but essentially see that we looked at over 200 fish of 18 species, and none of them had Dracunculus in their tissues. We also looked at frogs across these 5 different trips. We sampled multiple species of frogs. We found Dracunculus medinensis in 2 of them. And through surveys of people, we found out that people were eating 2 of these species of frogs as well. We didn't find infections in the giant bullfrog, but you can see on this table we only sampled 3, so that doesn't really mean much for not finding the parasite. So what we found during these field studies was essentially a lot of nematode larvae from 276 amphibians. But only 10% of them really looked like they could be Dracunculus when we looked at them under the microscope. And of those, we confirmed that 7 were Dracunculus medinensis. And those 7 larvae were found in 4 different amphibians. So not a whole lot of amphibians were infected, and none of the fish. So does this really support the number of infections we're seeing in dogs and cats in Chad? Of course, that's the big question that we're still working on. And the fact that we didn't find it in any of the fish was quite interesting because we sort of expected to see some fish infections. Concurrent with that field work, we were doing experimental work back here in Athens, where we were taking fish and frogs and exposing them to Dracunculus to see if they were experimentally susceptible. So on this slide, you can see that there were multiple species of frogs and a toad species that we exposed to either Dracunculus insignis, which is our native North American species, or when we had parasites available for Dracunculus medinensis, we would use that parasite. But regardless of the species of amphibian, they were all susceptible. We did a long-term study in African clawed frogs, which are a fully aquatic species that's endemic to Chad, and we saw that we could maintain infections with Guinea worm or with Dracunculus larvae in them up to 8 months. Just to show that those parasites that we were retrieving from these amphibians were actually infectious to a definitive host, we fed those parasites to ferrets, and they did become infected, confirming they were infectious. Of course, we're interested in fish as well, so we were exposing several different species of fish. And so these 4 that are shown here on this slide, 2 of them are native to Chad, and in all cases, they were negative for any parasites in their tissues. However, when we looked at Centodontis catfish, also native to Chad, we found that 3 out of the 4 fish that we exposed to infected copepods actually became infected with parasites in their tissues. So there does appear to be some difference in susceptibility of fish for Dracunculus. During those studies, though, we noted that there were some species or some individuals that just did not eat a lot of copepods. And so while our initial goal was to determine whether or not they were susceptible, if they are susceptible but don't eat copepods, then it's not really relevant to the natural history of this parasite. So we did another study looking at copepod ingestion rates. And so we had a bunch of different species that you could group into fish, or adult aquatic frogs, which would be xenopus, newts, and then tadpoles of a variety of different amphibian species. And you can see in this slide that fish ate the vast majority of copepods that were provided to them during a 24-hour period. The adult xenopus frogs also ate a large number of copepods, but less than the fish. And then the newts and amphibians ate copepods, but not very many. So when we broke it down by species, we could see that there were some species level differences in fish in terms of the number of copepods that they ingested, but regardless of the species, they tend to eat a lot. Whereas with the amphibians, when we looked at the different species of tadpoles, there wasn't a lot of variation, although there was some. But generally speaking, they ingested far fewer than the fish. So these experimental studies showed that at any given time, a large number of the fish would have lots of copepods in their gastrointestinal tract because they would consume nearly all of the copepods provided to them within a 24-hour period. And so what that suggests to us is that maybe they could be serving as a transport host, and I'll talk to you more about that in a minute. But specifically for the frogs and the tadpoles, they also ingested copepods, but they were ingesting far fewer and more slowly. But we knew, based on our experimental trials, that a large number of these frogs would become infected. One big difference between those 2 studies, though, was that in the copepod ingestion trial, we only gave them 24 hours to ingest them. Whereas with the susceptibility trial, they had at least 72 hours during which time they could ingest copepods. So we had a large number becoming infected. So obviously they do over time consume enough copepods to become infected. So we still think that they could be important paratenic hosts for this parasite in nature when they would have more time to ingest copepods. So again, let's go back to the fish. So all the experimental studies, the field studies really showed that it was difficult to find larvae in the tissues of these fish, at least for all of the species except for the one species of catfish, the Synodontis. But the thought was maybe they could be serving as transport hosts. And the difference here is just simply fish are consuming large numbers of copepods. Some of those copepods could be infected. And at any given time, those parasites are transiting through the gastrointestinal tract of fish. If a dog or a person or someone else ingests that fish, during the time that those parasites are transiting through, they could potentially become infected. So the fish would just simply be transporting the parasites to them. So we did an experimental study to look at this. We fed fish to ferrets within 2 to 3 hours of allowing those fish to ingest Dracunculus infected copepods. And you can see on this slide, on the right-hand side, 3 out of 4 of those ferrets became infected after eating those fish. So we confirmed that, in fact, fish could serve as transport hosts. So now we have both experimental and field evidence suggesting that there's some role for fish and frogs in transmission, but how important is this role? The short answer is we don't really know. But there have been a couple studies that have tried to look at this. One modeling study looking at the potential role of frogs showed that they could be important if they ingest copepods and then are eaten by dogs. But on the other hand, they could be important for actually suppressing transmission if they're ingesting large numbers of parasites in the environment, but then a large number of them are not being ingested by dogs. So the problem is we don't know how many frogs or tadpoles a particular dog would eat in a given year. So it's really hard to gauge how important of a role that may play for transmission. We still believe fish could be important. And in fact, when looking at risk factors for villages that have infected dogs, the fact that those are fishing villages really come out as a strong correlate. Looking at human infections, folks have tried to correlate whether or not fishing or eating fish is related to infections, but the vast majority of people do eat fish. So it was really hard to tease that out. And given the small number of human cases compared to dog infections, really makes it difficult to determine risk factors for humans based on fish alone. So with that summary of some of the work that we've done in the past, I just want to talk about some of the current and future work that we're doing. One is an interest in how are cats getting infected. One way we did this was just simply look at what cats were doing. So we installed little kitty cams on their collars, and you can see here is a picture of a cat eating a beetle. And you can see another cat here eating on something, and then it starts to eat here, and you can see that that's a fin of a fish. Here you can see a cat carrying a gecko. We also had video of a cat carrying rodents quite commonly. Geckos and rodents both are hit hard by cats and Chad. Here you can see a cat eating on something, a big old chunk of meat. And if you look closely, you can see there gill fins and vertebrae of fish. So this cat was having a very nice meal of fish. So we certainly have some good evidence that cats are ingesting fish and fish products based on this camera data. And so we're going to continue trying to understand how these particular cats may be infected. And then here, cat drinking water. So there's the potential that they actually ingest some copepods as well. Another big interest of ours is what is the role of wildlife in this natural history and transmission. And so we have studies going on with camera traps in various villages with and without guinea worm infections to look at the interactions of wildlife with those villages. So what wildlife are surrounding the villages, what wildlife species come into the villages, which wildlife species are using water bodies in and around the villages, and what interactions do these species of wildlife have with domestic animals, with the goal of just trying to understand if wildlife are actually infected with guinea worm, what risk may they play to contaminating the environment around a village. And then we're also looking at flubendazole as a potential treatment or preventative for guinea worm infections in dogs. So from 2019 up until last year, we did a field trial in Chad using flubendazole treatment of 435 dogs, 3 different treatments, 6 months apart, to determine whether or not flubendazole could either treat current infections or prevent infections during that time frame. We didn't find a lot of evidence for any effect of that drug on either worm emergence or larval viability, but concurrent with that field study, we were doing work with ferrets in the laboratory. And in that particular study, we infected ferrets, and then the idea was that we were going to treat them with 3 rounds of treatment 6 months apart, same as the dogs. But because of a delay in determining whether or not ferrets were infected or not, because not all exposed ferrets will become infected, and so we didn't want to treat uninfected ferrets, we didn't detect infections in ferrets until about 9 months into the study. So at 9 months, ferrets were given a treatment, and then a month later, they were given another treatment, just 30 days apart between those 2 treatments. And so in that particular study, we found that there was significant damage to the worms, to the worm uterus, and to the development of the larvae, to the point where there's any larvae that were actually present were incapable of infecting copepods. So that gave us great hope that there is some effect of flubendazole, at least on the development of larval stages within infected hosts. And then the big difference between these 2 studies, we believe, is the fact that the ferrets were given 2 doses so close together, which actually, in effect, gave them a higher dose because this drug is persistent for longer than a month. And so what we're doing is we are revisiting this study with a new field trial in dogs in Chad, where we're enrolling 2,000 dogs, half of which will be infected with a higher dose of flubendazole, and the other half will be a control arm. And we will look to see if this drug is useful in lowering or eliminating worm emergence or larval development within those hosts. The data that came out of the ferret study was really suggestive that it may work. So we're excited to see where this leads us over the next couple of years. We've also been active working on developing and validating and implementing new diagnostic tools. This first one here is in collaboration with the Centers for Disease Control and Prevention, who developed a serologic test for humans, and then they adapted that test to work with dogs. And so now the idea would be we would have a serologic test that we can use to test dogs in a village to see whether or not they have developing guinea worms in their system. Right now, the only way that we can tell a dog is infected is to wait for a worm to emerge. Well, once a worm emerges, there's a risk that that dog has contaminated a water body. And if you don't know what water body that is, then there's no way that you can treat it with a bait to kill the copepods. So what folks are working on is a way to develop an antemortem test in these dogs to determine whether or not there's a developing parasite in them so that they could be tethered or at least not allowed access to water when that worm may begin to emerge. And so the results of this serologic test are quite promising. Antibodies are detected well before worms are expected to emerge from them. Other diagnostic tests that we were working on is rapid field deployable diagnostic tools, molecular tools. One is a LAMP assay that is similar to PCR and real-time PCR in that you have amplification of the DNA, but it's done in a machine that doesn't require a significant amount of resources in the field. As an adjunct to that, we've also developed a real-time PCR that is quite useful for confirming whether or not a worm is guinea worm or not. And we're currently working on ways to make that real-time PCR field deployable as well. So the nice thing about these 2 assays is that you could deploy them to the field and do testing of materials in the field and know very quickly whether or not a copepod is infected with guinea worm, or if a guinea worm that you removed from an animal is actually guinea worm. Taking tissues from frogs and fish, taking intestinal tracts from fish, testing those for guinea worm larvae, And so we're really excited to start to deploy these into the field and see how they work. So in terms of the life cycle of this parasite, 150 years ago or so, folks determined that copepods were the intermediate host, and ingesting those copepods led to infection of humans. And for most of that time frame, humans were the predominant or only host for this parasite. Clearly, though, based on all these studies in recent years, we see that this system is a bit more complex than that. It's not just simply a human drinking water and ingesting copepods life cycle. There's the potential for fish serving as transport hosts. There's the potential for amphibians transmitting infection to a definitive host. We have dogs that are infected. And so the system is just simply not a human copepod system anymore. There are other players. And so that is the interesting part about the system. It's also the frustrating part about the system. As with any research, you usually answer one question and you come up with 3 or 4 other questions. So, of course, we have our long list of new questions that we continue to look at, and hopefully will continue to contribute useful information to this guinea worm eradication program. Talks like this tend to be quite doom and gloom, especially when you're talking about eradicating a parasite that's so important to human health. And it has had some stumbling blocks over time with the emergence of this parasite in dogs. But I guess if we want to sort of either think about the light at the end of the tunnel, or maybe hope that things will get better soon, we actually do have some data that suggests things are getting better now. If you look at this graph, 2019 numbers of cases in dogs are shown in blue. 2020 is in green. And then the number of infected dogs per month is shown in red for 2021 up until August of this year. And you can see each one of those years, the peak of the number of infected dogs has decreased dramatically during the peak transmission season, which runs somewhere between March and July. And so the numbers tend to level off and even out, at least for 2019 and 2020, during the low transmission season of October, November, December. But the bulk of the worms that are entering into the environment and infecting copepods, which then could lead to more infections in dogs, cats, wildlife, or humans, occur during this middle part of the year, and those numbers are decreasing each year, which is fantastic news. So we certainly hope that this trend continues in terms of the numbers of infected dogs, with the hope that once it goes away in domestic dogs, it will not be sustained in wildlife. It won't be sustained in cats, which right now do represent a fairly low number of infections compared to dogs. And with that, this parasite will disappear from humans. So with that, I would just like to take a few minutes to thank many different groups. One of the most important groups would be the communities that welcome us into their community to work with them to help eradicate this parasite. And in many cases, do research to understand transmission of this parasite. The ultimate goal is eradication, but at the same time, we're trying to gather useful information in a research framework that can help inform new interventions that will ultimately lead to the eradication of it. Stateside, there's a large number of people that are involved in these studies. So the copepod ingestion trials, the experimental infection trials of fish and frogs and other hosts are incredibly time consuming. And so there's an army of people that have been helping with this project for several years now. And those include high school students, undergraduates at UGA, graduate students, a whole group of technicians, and postdocs. There's also other faculty at UGA that have been involved in various aspects of this research. And so I certainly can't individually thank them all, but again, there's a huge number of people that have helped contribute. And so that's just at UGA. And so moving beyond that, I have to give a huge thanks to the Carter Center, not only for funding the research that we do, but taking the lead on this guinea worm eradication program. There are a huge number of people involved with this program, as you can imagine, at the Carter Center, both here in the United States, but in country as well. The World Health Organization, folks that are present within the Chad Ministry of Health, within the Guinea Worm eradication programs in country. I want to thank all of the funders and donors that give money to the Carter Center that allow us to continue this work. Different aspects of this work was done in collaboration with the CDC, and so there's a large number of people at the CDC that have worked with us on these projects, and I certainly appreciate their help, and I also appreciate them coming to us to bring us into some of the studies that they are proposing as well. John Bryan is a fantastic veterinarian and pathologist who's been working with us on a variety of projects, primarily in Chad. Charles McKenzie and Tim Geary, who are phenomenal partners with the Guinea Worm treatment program with the flubendazole trial. And so we can only hope that we'll get some fantastic results from that, and combined with what we're seeing in terms of decreasing numbers of dog infections, will make this presentation a very different presentation in the future. And we'll actually be talking about how we had a successful eradication program. With that, I would like to thank you for your attention. Here's my contact information. Normally, this is the time I would take questions, but please feel free to reach out to me if you have any questions or comments about it, and I'd be happy to talk to you. And if you would like a really nice overview about some of the work that's being done in Chad right now to try to eliminate transmission within dogs and some of the challenges with that. A couple of years ago now, the New York Times had a really interesting article they put together. So again, thank you very much.
Thank you, Michael, again for the great presentation on guinea worms. So we already have audience post questions in the YouTube chat. The very first question I have is from Kyle. Is deliberate infection of dogs with guinea worm a concern, seeing that there is a financial reward for turning infected dogs in?
Yeah, that is certainly a concern. We have not seen any evidence that people are doing that. And so, yeah, certainly we hope that people aren't going to turn to that. The financial reward, though, is something that really helps engage people and get them to help bring infected dogs in so that we can help prevent transmission into the environment.
Thank you. For the next question, it's from Diane Burton. When you were looking at the frogs and fish, were you looking at the whole animal, the internal organs, or the flesh?
Yeah, so when we're looking at fish and frogs as potential paratenic hosts, what we're looking for are actually those L3 larvae in their tissues. So I was vague when I was presenting, but essentially when we euthanize an animal, we necropsy it, and we pull the gastrointestinal tract out and set that aside. We don't really look at that, at least early on, because there's tons of parasites that are going to be in there, and what we're looking for is guinea worm. And then we take all of the internal organs, the liver, pancreas, heart, etc., and we put that into a separate petri dish. And then we take all the flesh and we mince that up and put into a separate petri dish as well. So we examine both the internal organs and the flesh, the tissues, to see if there's parasites there. And essentially what we do is we mince it up really small in a petri dish, and we sort of examine it immediately to see if we see any parasites coming out of the tissues. And these are, of course, L3 larvae, so they're very, very small. We're looking at everything under a dissecting scope. And then if we don't see anything, we let those sit over time and then examine them again. In the field, we actually set up these Behrman funnels. So all parasitologists know what a Behrman's is, but we, instead of using it to look for parasites in feces, we actually take all of those tissues or the organs in two separate Behrmans and set it up so that those larvae will come out of those tissues and fall to the bottom of the tube. And then we can actually look in the petri dish for those larvae. And as you saw from the talk, we get a ton of larvae. And then we have to sit there and put each individual larvae onto a microscope slide to look to see if it potentially looks like it could be guinea worm. And from there, we move on to molecular work to identify them. Going forward, we have a particular interest now in looking at the actual gastrointestinal tract, at least of fish, because of the thought that maybe they're serving as transport hosts. So whereas before we sort of ignored the GI tracts, even though we knew there was tons of copepods there, we were much more interested now in looking to see if we can detect using either that LAMP assay or field deployable real-time PCR to see if we can actually find infected copepods within the GI tract of those fish.
Thank you. And our next question I posted in this chat here is from Dr. Matt Bollick. He said, great talk. I know you did not talk about this species very much, but any idea on how Dracunculus globocephalus, which infected sleeping turtle, releases larvae into the external environment?
Yeah, I didn't have enough time to talk about all the reptile Dracunculus species, but I'm really interested in those parasites for sure. One from just an evolutionary standpoint. Pretty much everywhere people look in the world in snakes, they find Dracunculus species. But for mammals, there's only a few species and they're much more restricted in their range. So from an evolutionary standpoint, it looks like reptiles probably are the ancestral host for this group of parasites. So it's really a cool group to look at. There's some old literature that actually also looked at the ability of tadpoles to become infected with Dracunculus ophidinensis from a snake. So there was yet another example of some of this older research that showed amphibians could be important, which was really helpful. In particular, globocephalus and snapping turtles, we've looked for that parasite quite a lot. And we haven't found it in snapping turtles or any of the other aquatic turtles that we've looked at in our area. It seems widespread in the United States, so it's probably just a matter of we've been unlucky to find it. But in terms of how the parasites get out, I suspect it's similar to all of the other mammal species in that the female is in the subcutaneous tissues and forms a blister or a nodule and then emerges. And in fact, recently we've gotten more into the snake side of things. And so we've actually had some aquatic snakes submitted to us that had nodules in their skin. And when we teased those nodules open, there were actually coiled up worms inside, and those ended up being Dracunculus. So I suspect it's similar, but I've certainly never observed it in a snapping turtle. And so I don't really know.
Thank you. And our next question is from Miriam Kelly. Has there ever been a CAP study on guinea worm in the villages you complete research in?
Yeah, so great question. I have not personally been involved in any of the knowledge, attitudes, and practices surveys that have been done in the various countries where guinea worm is endemic now or historically was endemic, but there has been lots of research on that. Mainly in trying to not only understand knowledge and practices related to guinea worm transmission or potential risky factors for transmission, but just understanding the most appropriate way to educate a group of individuals about how they can prevent transmission, because each country is, of course, going to have a different practice in terms of understanding the way to break the life cycle. And so there's been lots of work on the human dimension side for sure. I just haven't been involved in that work.
Thank you. Our next question is from Mickey. My impression on the eradication program has been on the human copepod interface. Has there been any insights onto the copepod larva parasite side of things?
Yeah, and so a standard practice historically and currently is to use a bait to kill the copepods within a given water body if there's a suspicion that either a human or a dog has gone into that water body with a worm that's emerging. And so if there's any indication at all that a water body potentially has guinea worm in it, they will treat with a bait and then do repeated treatments with a bait to make sure that copepods within those water systems are clean. The other big thing that they try to do, of course, is provide safe drinking water options, but not everyone's going to have that option. Not everyone's going to have the filters necessarily all the time. So they do really focus in on the human copepod side of things, either manually removing copepods when drinking or chemically removing the copepods from these high-risk areas. And in some cases, if you've just got cases within a village, but you don't know whether or not a water body has been contaminated, Ideally, you would have an aggressive campaign in place to treat those water bodies with a bait to ensure that there are no copepods available. Sort of the good news is it takes about 10, 14, 16 days or so for these parasites to develop to the infective stage within copepods. So you have a small window of time where you can go out and treat these water bodies. In terms of looking at the copepod larval side of things, I think the best bet is targeting the copepod itself, just simply because if you don't have copepods, you're not going to have any of the developing parasites there. I don't know of any treatment options necessarily that you could put into the environment that would kill selectively the nematode within the copepods or within the environment before they get into the copepods.
Thank you. Our next question is from Kyle. Here. Do you have an opinion on the cost versus benefits of guinea worm eradication? Considering that there are very few human cases each year, do you think the arguments that our limited resources could be better directed to a more prevalent disease are? Yeah, thank you.
Yeah, and so this is a very large program, the guinea worm eradication program. But one thing that it does really well is capacity building. And so it is building resources in country that's not only going to impact guinea worm, but other diseases as well. And so that is one positive aspect of this program. So looking forward, hopefully we do see human cases continue to decline. Hopefully we'll continue to see dog cases decline as well. But I think there's grander benefits to this program other than just simply eradicating guinea worm.
Thank you. And our last question is from Mickey. Beyond companion animals, are there any concerns about Dracunculus on domestic livestock?
Historically, there have been rare reports of Dracunculids in cattle and horses in different endemic countries. But those were very old reports, and there are no contemporary reports of that right now, at least in the few countries that have endemic transmission. So I don't really know. I suspect it would be rare to have those types of infections, and I don't suspect that they would be responsible for maintaining any sort of endemic transmission within the environment if they, in fact, do become infected. One thing to point out, all of these parasites that emerge from people, dogs, cats, and other animals, there is a program in place to collect those worms and have them properly identified. And a good number of those parasites that do get collected are not guinea worm. And so when you look back at some of these older studies that talk about worms emerging from the skin of certain animals, you have to think there could be other things, other filarial worms that would be present there that could have been confused with guinea worm. So right now, there's no evidence that they're involved in the life cycle.
Thank you. Thank you so much. I think that's all the questions we have for this section.